Could the cure for genetic diseases lie in the very fabric of life itself, our DNA? This complex, double helix structure, a precious inheritance from our parents, is a fragile masterpiece comprising 3 billion letters. It's a blueprint, a code, that holds the secrets to our individuality. But like all things, it's not invincible. Factors like sunlight, smoking, and even the spontaneous errors of our own cells can inflict damage, leading to what we call point mutations. Point mutations, often just a single letter swap in the DNA sequence, are the most common type of genetic damage, and sometimes these seemingly minor changes can trigger devastating genetic diseases. Think of sickle cell anemia, progeria, muscular dystrophy, all born from the intricacies of our DNA. It's a daunting reality, but one that propels us forward into the realm of possibility and innovation. Yet, hope lies on the horizon with the advent of base editing. CRISPR technology, a breakthrough in genetic editing, has its limitations. Now let's delve deeper into the world of CRISPR. A revolutionary tool in the field of biotechnology, CRISPR, or clustered regularly interspaced short palindromic repeats, allows us to make precise cuts in DNA at specific locations. Imagine it as a pair of molecular scissors guided by a GPS that can snip away at our DNA strands. However, like all technologies, CRISPR is not without its challenges. Picture our DNA as a long, intricate novel. While CRISPR can help us remove whole chapters or even just a single paragraph, it struggles when it comes to correcting individual letters or words known as point mutations. These point mutations are the most common type of DNA damage and can sometimes cause genetic diseases. When it comes to genetic diseases like sickle cell anemia, progeria, and muscular dystrophy, which are caused by these point mutations, CRISPR's capabilities fall short. Cutting an already mutated gene won't restore its function. It's akin to removing a misspelled word from a sentence. It doesn't automatically correct the sentence. Moreover, introducing new correct DNA sequences to replace the mutated ones is a complex task. It's not as simple as pasting the corrected word into the sentence. Every cell type presents its unique set of challenges, making this process even more complicated. So while CRISPR has been a game changer in the realm of genetic editing, there's a need for a more efficient solution, especially when it comes to correcting point mutations. This is where base editors come into play. Enter base editors, the next generation in genetic editing. These molecular machines have the potential to directly convert one DNA base to another without disrupting the rest of the gene. It's like having the power to correct the spelling of a word without altering the rest of the sentence. But we'll delve more into base editors in our next scene. Stay tuned for more on this exciting development in the world of genetic editing. Base editors, the molecular machines designed to rewrite DNA. These ingenious devices have been crafted by researchers to tackle the challenge of point mutations in our DNA. But how do they work and what makes them so special? Base editors are a fusion of two key components: a programmable CRISPR system and enzymes that can chemically modify DNA bases. This unique combination allows them to directly convert one DNA base to another without disturbing the rest of the gene. This is a significant step forward from the traditional CRISPR technology, which involves cutting the DNA and can be quite disruptive. There are two types of base editors, each capable of different conversions. The first type can change a cytosine, C, to a thymine, T, and a guanine, G, to an adenine, A. The second type, on the other hand, can convert an adenine, A, to a guanine, G, and a thymine, T, to a cytosine, C. This second type of base editor is particularly fascinating, as it was developed by evolving a protein in a laboratory to perform the essential chemical reactions. It's a testament to human ingenuity and our ability to harness the tools of nature to our advantage. So why is this so important? Because base editors have the potential to correct the point mutations that cause many genetic diseases. They offer a new and efficient way to fix these errors at their source without causing additional damage to the gene. In just 3 years, base editors have been widely adopted by researchers around the globe. We have witnessed over 6,000 requests and 100 published papers on the subject. They have been used to correct point mutations in animals that cause human genetic diseases like progeria, anemia, and muscular dystrophy. Even in the field of plant science and cancer research, base editors are making waves, introducing beneficial changes and helping to explore the roles of individual gene mutations. The adoption and application of base editors are rapidly growing, and as they continue to evolve, their potential seems limitless. These molecular machines are not only rewriting DNA, but also rewriting the future of genetic disease treatment. Base editing, though promising, still faces challenges. As we stand at the threshold of a new era in biotechnology, we must recognize the hurdles we need to overcome to fully harness the potential of base editing. The first major challenge is delivery. How do we get these molecular machines, these base editors, into human cells where they are needed? The human body is an intricate system with numerous checks and balances. It's not as simple as just injecting base editors into the bloodstream. We need to find a way to guide these editors to the right cells without triggering an immune response or causing other unintended effects. The second challenge lies in precision. Base editing, while innovative and powerful, needs to be accurate. The goal is to edit the specific point mutations causing disease and nothing else. Off-target editing could lead to unwanted changes and potentially new problems. This calls for further refinement of the technology to ensure its precision and safety. But, as with any scientific endeavor, these challenges are not insurmountable. They merely represent the next steps in our journey of discovery and innovation. Continued development of new base editors, harnessing the power of artificial intelligence, and collaboration among scientists, doctors, ethicists, and governments are all crucial. By working together, we can ensure the safe and ethical application of this transformative technology. The potential of base editing is immense. It promises a future where we can not only pass on our DNA to our children, but also the means to protect and repair it. A future where genetic diseases like muscular dystrophy, progeria, and sickle cell anemia are not inevitable, but treatable or even preventable. Imagine a world where we can correct the genetic typos that cause disease, where we can rewrite the code of life to improve health and well-being. This is the future that base editing holds. This is the future we are working towards. With base editing, the future of curing genetic diseases by rewriting DNA is within our reach.